Okay. So for urinary system, we're going to talk about the gross anatomy of the kidney and its coverings. We'll talk about blood supply through the kidney, the anatomy of a nephron. We'll talk about the three major processes of the urinary system, describe the forces or pressures that promote or counteract glomerular filtration. We'll talk about how we control glomerular filtration rate, or GFR. Then we'll talk about the mechanism underlying water and solute reabsorption from the renal tubules and peritubular capillary, and describe how sodium and water reabsorption are regulated in the distal tubule and collecting duct. So for the urinary system, you're actually going to have two trace a drop pathways, one for blood and then one for filtrate/urine. So your urinary system is composed of your kidneys, which are going to do all the filtering and reabsorbing and secretion. And then after urine is made, they're going to travel down the ureters to the urinary bladder to be stored, and then out the urethra. Okay. During micturition, which is urination. There's a different word for urination. Micturition. So urinary system functions. The urinary system is going to regulate the total volume of water in the body. This is under hormonal control. Anybody remember the two hormones that are going to regulate water movement? ADH, and what was the other one? I heard it. Aldosterone. And remember that ADH is for water reabsorption. And aldosterone is for sodium reabsorption, but water follows it. All right. The urinary system is also going to regulate total concentration of dissolved solutes in the bodily fluid, so it's going to maintain osmolality. We're going to have regulation of concentrations of individual ions in the extracellular fluid, like potassium. Potassium regulation is related to aldosterone. So aldosterone is for sodium reabsorption, so putting more sodium in the blood. So how aldosterone works is it actually uses a sodium-potassium pump. So every time it's pumping sodium into the blood, it's pumping potassium in the opposite direction towards the urine. So while we're regulating sodium levels, we're also regulating potassium levels at the same time. Okay. What is the hormone that regulates calcium concentrations? Parathyroid hormone, or PTH. The urinary system is also going to ensure we have an acid-base balance, so the correct amount of hydrogen ions and bicarbonate ions to maintain our blood pH. What should our blood pH be? Yes, 7.35 to 7.45. We're going to excrete metabolic waste like acids and urea, which if you haven't finished that nutrition lecture, you'll learn about urea and protein breakdown during the rest of that nutrition lecture. And then also getting rid of foreign substances like drugs and toxins. The kidneys, as you should remember, produce erythropoietin when oxygen levels are low, and then also renin when blood pressure is low, so we'll bring back that renin-angiotensin-aldosterone pathway again. And then it's responsible for converting vitamin D in its active form. So whenever we're making vitamin D, what is the trigger that starts that whole pathway? You learned about it in B32. Huh? Something in your external environment. Sunlight, UV rays are going to start the process of making and activating vitamin D. All right, any questions about urinary system functions? Okay. So the kidneys are a retroperitoneal organ. So that means they lie between the dorsal wall, the very back wall of the abdominal cavity, and the peritoneum is in front of them. So they're only covered by membrane on the front and lateral sides of the kidneys. All right. So hopefully you guys put retroperitoneal during the practical yesterday when talking about the kidneys. All right, the kidneys extend from the T12 to the L3 vertebrae. The right kidney is slightly lower. And why? What organ takes up a lot of space in the upper right portion of your abdominal cavity? The liver. So the right kidney is slightly lower than the left because of the liver placement. So it makes room for the liver. Or accommodate the liver. All right. So more about the anatomy of the urinary system. So there is something here, a passageway here called the renal hilum, which is why I told you when we talked about the hilum of the lungs, you want to say it's the hilum of the right lung or hilum of the left lung, because now we have this renal hilum too. So you want to know the difference between hilum of the kidneys versus hilum of the lungs. It's the same thing. It's a passageway for tubes and nerves and vessels. Okay. So it's a vertical cleft on the medial side of the kidney, and it's going to allow entry for the ureter, vessels, lymphatics, and nerves. And we also have some supportive tissues. We have the renal fascia, which the renal fascia is going to anchor the kidneys in place, the thick connective tissue. We have this perirenal fat capsule, which is mainly for cushion. And then the fibrous capsule on the very outside is going to prevent the spread of infection. So one clinical application about this fat capsule or cushion is that, I don't know if you've heard this, but healthy weight loss is about 1 to 2 pounds per week if you're trying to lose weight. Dramatic weight loss can cause you to, and if you're losing weight too fast, it can cause you to lose that fat capsule. So dramatic weight loss can lead to loss of the fat capsule, which can cause the kidneys to droop. So instead of the kidney looking like this, it will drop lower. And you may get a kink in that ureter because it has dropped. Okay. This kidney droop is called renal or nephrotosis. And ptosis is P-T-O-S-I-S. So either renal ptosis or nephrotosis. So that kidney droop can lead to a kink in the ureter. And if we get a kink in the ureter, just like we get a kink in a water hose or such, the flow is going to stop. And what's going to end up happening is the urine is going to backflow into the kidneys. So we get a backup of urine, which when you have a backup of urine and it causes swelling in the kidneys, it's called hydronephrosis. And depending on how severe it is, it's going to determine the treatment. So if your symptoms like flank pain and such, infections, things like that, if it's very mild to moderate, then they may just kind of wait it out and provide antibiotics if you were to get an infection. But if it's really severe and your symptoms are severe, they may have to go in and do surgery to reposition the kidney in the correct place. So any questions about renal ptosis or hydronephrosis?
Professor, so renal ptosis is a kink in the ureter, and hydronephrosis is back of the urine?
And hydronephrosis could be swelling of the kidneys due to the backing up of urine, or it could be due to an infection. Hydronephrosis, more generally, is just the swelling of the kidneys. Yeah.
This is a general question, because I always feel like wrestlers have to kind of lose weight rapidly. So, is this common in that type of field, or do they have like coaches to regulate it?
I have to look in to see if it's more common there, but anybody who's experiencing dramatic weight loss, more than the 1 to 2 pounds a week, are going to be at risk of something like this. I don't know if they're studying that and looking into that, so I can look into it though.
The same thing could apply to like Ozempic.
Yeah, Ozempic, yeah, and Wegovy and all of that. They already know it causes bone mass loss and muscle mass loss as well. It's just it's so new, so we're not really sure of the long-term effects just yet, which I would assume probably that too. All right, so your kidneys have 2 layers, which you should have seen during the pig dissection. We have the renal cortex, which has blood vessels to feed the structures called glomeruli for urine formation. And then the renal medulla, the blood vessels are going to carry blood, and the tubules are going to carry filtrate in that region.
All of this is being recorded.
Huh?
Recorded, right?
Yes, being recorded, yes. I promise. All right, so the structures that we have within the kidneys are the medullary or renal pyramids. You can call them either one, but it's just that they're organized in these little triangles, these pyramids. So again, you can call them medullary pyramids because they're in the medulla of the kidney, or you can call them renal pyramids, either one. And then we have minor and major calyces. If we're talking about just one, it's called a calyx. And so how it works is once we make urine, they're going to travel from these minor calyces here, so these small tubes, and they're going to lead into major calyces, which are larger areas. And then those major calyces are going to lead into the renal pelvis, which is this area where they all come together, where all of that urine comes together. So minor calyces lead to major calyces, which lead to the renal pelvis. And then once the urine moves into the ureters, then the urine is going to move down that tract through peristalsis, just like it did with chyme and feces and such in the digestive system. Any questions on those structures? Okay. The kidneys receive 25% of cardiac output for urine formation, which the word for urine formation is diuresis. So make sure you write that down somewhere. Urine formation is diuresis. And then once we get to the very end of urinary system and we talk about like actual excretion and urination, that's called micturition. But the forming of urine is called diuresis. So we learned with the digestive system, how much of cardiac output does the digestive system get? 25% also. What is that circulation called? Splatchnik. Okay. At the end of these renal arteries and veins here in this section here, we have something called a renal plexus. And that renal plexus is going to contain autonomic nerves and ganglia. And I think there's a note about this in your lecture notes already. But it's going to have mostly sympathetic fibers. Because if you remember, your sympathetic system is the one that's responsible for regulating blood pressure, called sympathetic tone. So mostly it's going to be sympathetic fibers that are controlling the blood pressure at the kidneys. Why does it keep doing that? Because this board's in the way. You see me walking around like this today, just mind your business. No, I was lifting yesterday and I used a 55-pound dumbbell for like squats and lunges. And I think I need to wear a belt anytime I do legs, I feel like, because it's just, I'm getting old. It's very sad. I used to be able to back squat like 225 pounds. And I want to get there, but I don't think my back can take it anymore. Thank you. Huh? Oh yeah, I know. I didn't wear it yesterday. I was like, it's just a single dumbbell. I'll be fine. So, yeah, I will. All right. Any questions about that? So the renal plexus is just the gathering of the nerves and ganglia that are going to control blood pressure and blood flow through the kidneys. All right. So I did say you have tracer drops. So you're going to have to be able to trace a drop of blood through the kidneys, as well as tracing a drop of filtrate through the kidneys. Now, this is a really intense, long pathway. You don't have to know all of these. Ooh. It's still recording. Okay. So you don't have to know all of these. So I'll get rid of that one, and 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 that one. So these are the only ones you have to know. So there is a little space for you to do a tracer drop, I think, in your lecture notes. Yeah. So you go from the aorta, which is ultimately bringing the blood in from the heart, and then it's going to approach the kidneys at the renal artery. From the renal artery, we're going to approach these filtration structures called glomeruli, and a single one is called a glomerulus. So the vessels leading into the glomerulus are called afferent arterioles. Then the blood goes through the glomerulus and exits through the efferent arterioles. Now, on the next part of that tracer drop, you can put, the one we're going to deal with the most is going to be peritubular capillaries. So underneath that, you can put or vasorecta, and I'll explain the difference between those two in a second. And then as we exit out of the kidneys, we're going to exit out through the renal vein and then eventually go to the inferior vena cava back to the heart. So I'll give you a second to write that pathway down.
Professor, where do you say to read from this?
So right peritubular capillaries in that spot for peritubular capillaries, and then right underneath that just put or vasorecta. I know the arrows and lines are a little bit off. I didn't realize that it happened that way when I printed it. But you can make room. Yeah, put peritubular capillaries. That's the one that we're going to deal with the most. And then underneath it, you could put or vasorecta. So basically, we have two different structures that are going to be filtering our blood. They're called cortical nephrons and juxtamedullary nephrons, which Professor Green didn't teach you, so you're going to have to learn it now. Cortical nephrons are more for regulating solutes in the urine, while the juxtamedullary nephrons are more for concentrating urine, so more about water reabsorption and secretion. And so peritubular capillaries are associated with the cortical nephrons, while the vasorecta is associated with the juxtamedullary nephrons, which we'll talk about in a couple slides here. Sorry, Gordon.
It's nice to learn this stuff.
Yeah. Okay, anybody still need this up? Okay. So the nephron is the smallest functional unit of the urinary system. Do you guys remember what the smallest functional unit of the muscular system was? Sarcomere. What was the smallest functional unit of your skeletal system? Starts with an O. Osteon. Osteon. What is your smallest functional unit of the nervous system? Neuron. Okay, just asking. All right, so the nephron consists of the renal corpuscle, which the renal corpuscle within itself includes the glomerulus. And remember, if it's plural, we call it glomeruli. So we have the glomerulus and the glomerular capsule, which is sometimes referred to as the Bowman's capsule, and I'm okay with either. But again, we're moving away from naming things after people. And so the glomerulus and the glomerular capsule are all held within the renal corpuscle. This is the site of filtration. I almost gave you guys today off and to do this online because of the exam and spring break, but this is too hard to have you learn on your own. And so this is where filtrate, which is pretty much pre-urine, is formed. So we call it filtrate as we're still making adjustments, adding things to the urine or the blood and vice versa. Once we're done making modifications, which is going to be after the collecting duct that I'll tell you about in a couple slides, then we call it urine. Okay, so when we're done making modifications, then it's called urine, but before that we call it filtrate. And then we have the tubules, where we have the proximal convoluted tubule. Remember proximal, when we learned about the body, it meant closest to the trunk. Proximal in this case is closest to the glomerulus. So the glomerulus is where we start and where all the filtration happens. And so the tube that's closest to the glomerulus is called the proximal convoluted tubule. And it's convoluted because the tubes are all kind of wounded together. Okay. Then we have the nephron loop, which you should see in your notes. It says that it includes an ascending and descending loop. So descending going down, ascending coming back up. And then we have the distal convoluted tubule, which is a part of the tubule that's farthest away from that glomerulus. And so the tubules are the site of reabsorption and secretion. So try to remember that it needs to be called reabsorption when we're talking about the kidneys and urine formation. Because in the digestive system, we called it absorption, when we absorb things from our food, because that's the first time that you've come in contact with that food, and then you break it down and you absorb it. In this case, we are filtering the blood, so there is stuff already in the blood. We filter that blood to make the urine, but sometimes there are things in that filtrate or urine that we still want to keep. And so then we put it back into the blood. And so because it was already in the blood, we filtered it out and then put it back. We call it reabsorption. So you have to make sure you call it reabsorption and not just absorption. Okay, does that make sense? Okay.
You said it's ascending and descending loops?
Limbs. Limbs, yeah. And I'll write that out in a second, yeah. So reabsorption and secretion are how we make adjustments to filtrate. Okay, any questions on this stuff? Who still need these notes up? Okay. All right, so this is a simplified version of a nephron. The tubes are going to be much more convoluted than what you see here. But here you have the renal corpuscle. This whole thing is the renal corpuscle, which includes the glomerulus, which the glomerulus is this ball of capillaries, and then the capsule around it. That's the renal corpuscle. Then we have the proximal convoluted tubule, which again will be much more convoluted in the models and the rest of the pictures you see today. But proximal convoluted tubule is the part of the tube that's closest to the glomerulus. And then we have the descending limb, nephron loop, ascending limb, so going down, looping, and then coming back up. This is the distal convoluted tubule, and that's all that's part of the nephron. This collecting duct here, it is not part of the nephron, so make sure you know that. It's not part of the nephron. And why it's not part of the nephron, it's a collecting duct for all the nephrons to feed into. Okay, so this comes after the nephron. All right. So let's do a trace a drop of filtrate together. And hopefully it doesn't erase and go away. So this is trace a drop of filtrate. So we're going to go from, and only talking about filtrate and not blood. So we can start at the renal corpuscle, which it's really going to be the capsule portion that filtrate's going to be in. And then from there, and I'll spell it out for you guys because you're going to have to spell it out in an exam. Proximal convoluted tubule. But the rest of this lecture, you'll probably hear me refer to it as the PCT. And then down the descending limb. Oh, that is not, oh yeah, it is. Is that how you spell descend? Yes, okay. Sorry, sometimes words look weird. Descending limb to the nephron loop, up the ascending limb, to the distal convoluted tubule, or DCT, to the, and now that's the end of the nephron itself, okay? And then from there, we go to the collecting duct, which a bunch of different nephrons are going to be emptying into, to a minor calyx, to a major calyx, to the renal pelvis, to the ureter, or aureter, to the urinary bladder, to the urethra, and then out the external urethral orifice. Now, as far as the descending and ascending limb, if I had you do like this entire pathway and you didn't include the limbs, I would be okay with that if you just went PCT to nephron loop to DCT. But those terms are in your learn list, and so I want to make sure that you know all of the structures in the order that they go in.
So we'd get extra points because.
Maybe. Like a quarter of a point, maybe. For each, maybe. We'll see. Did you guys attempt to spell out cholecystokinin for an extra quarter of a point? If you put Helicobacter pylori, I would have given you a quarter of a point too. Instead of H. pylori. What were the other ones? I think that was it.
Did not translate.
What's up, Mildred? a PCT to the nephron loop to the DCT, and then the collecting duct. But the collecting duct's not part of the nephron. So make sure you know the nephron goes from here to here. That's the nephron. The rest of it's not the nephron. Okay, anybody still need this up? Okay, you still remember this flow?
Yeah, but yours might be different from mine.
How is it different?
Like, we don't add the renal pelvis.
You didn't put the renal pelvis? That's insane. Include the renal pelvis, please. Okay. All right, everybody good with this before it goes away forever and ever? Okay. All right, so we have two different classes of nephrons, which this is going to be new for you, Gordon. We have what are called cortical nephrons. And you can tell a cortical nephron from this other one over here called a juxtamedullary nephron. You can tell the difference between the two by two different things. One, the cortical nephron is mostly in the cortex. This is the cortex of the kidney. Below the vessels here, this is all medulla. And so the cortical nephron is mostly in the cortex and has a very short nephron loop. The juxtamedullary nephron, what juxtamedullary means? Juxtamedullary means that it's right at the crux or right at the junction between the cortex and the medulla. Okay, so the juxtamedullary nephron is going to be across both sections. You'll have it in the cortex and the medulla. It's going to extend across both, and it has a long, very long nephron loop. And so the cortical nephron is going to do mostly reabsorption and secretion of solutes. So it's going to regulate what substances are going to end up in the urine, which are going to end up in the blood. While the juxtamedullary nephron is more for concentration of urine. If you're dehydrated, your urine is going to be very concentrated with not a whole lot of water. If you're overhydrated, your urine is going to be very dilute with lots of water. We'll talk more about that with urinary system too. Most of our nephrons are going to be these ones though, and I think it's already in your lecture notes. 85% of our nephrons are cortical. But that's mostly the difference between them. Their location, size of their nephron loop, and then the cortical nephron is mostly dealing with solutes, while the juxtamedullary is mostly dealing with water and the concentration of the urine. Got that? Okay. All right. So you'll notice, so the purple is the capillaries at both of these different types of nephrons. So the cortical nephron will have what are called peritubular capillaries, which again, this is what we're going to talk about the most with talking about movement of solutes. And then the juxtamedullary nephron has what's called the vasa recta, which is around the nephron loop, which is going to be for movement of water. Now, lucky for you guys, the concentration of urine and the movement of water is determined by something called counter current multiplication, which is not something we're going to cover at all in this class. If you take another physiology course in nursing school, you may learn about the counter current multiplication or multiplier, but you're not going to learn about it here. So we're going to talk mostly about that peritubular capillaries, not the vasa recta. Just know it for anatomy and the difference between the two. All right, any questions about those two types of nephrons? Yeah.
When doing the tracer drop, would you say, like, would you ever ask us, like, oh, do a tracer drop using a protocol?
No, if the tracer drop would be mostly for you would use the peritubular capillaries.
Okay.
If you did vasorecta instead, it would still be correct. I would still be okay with that. Yeah. And your next quiz, I think it's going to have a word bank anyway, so I think it'll be peritubular capillaries. All right, so this is just showing you a micrograph of these different glomeruli and then the peritubular capillaries that are around it. These are microscopic structures, so very small. This is a very magnified image here. All right, so the glomerulus produces filtrate while the peritubular capillaries reabsorb 99% of it. So we're reabsorbing 99% of what we filter out at the kidneys. We want to hold on to most of the water and solutes and just get rid of what we don't need. So there is something called a juxtaglomerular complex. So just like the juxtamedullary nephron meant that it was close to that connection between the cortex and the medulla, juxtaglomerular complex means it's going to be close to the glomerulus. Okay? So we have a juxtaglomerular complex, which are these set of cells here that are close to the glomerulus over here. And so the ascending limb, this is where the ascending limb and the afferent arteriole come together. So this right here is the ascending limb of the nephron loop. And actually, I'll go back so I can show you what that looks like. So, where am I at? Now, I think it's your book that says it's the ascending limb. But it should, let's see. Is that the proximal? Give me a second. Okay. But it also, so right here here's the where do I want to talk about it? Yeah. This is the juxtaglomerular complex here. Now, your textbook may say, I know they say it's like two different things about where it's actually located. Let me see. So the juxtamedullary complex, I think your book says it's the ascending limb. This would be the ascending limb, but the pictures make it look like it's more like the distal convoluted tubule. That really doesn't matter to me which one that you say it belongs to, because it all depends on the length of the nephron and all of that. So just know that the juxtaglomerular complex is where the afferent arteriole, which is the blood coming into the kidneys, is, or the glomerulus is, and then also just a part of the tubule. I'm not really concerned with what part of the tubule it specifically is, because I've seen different versions. But it's going to include 3 types of cells that are going to help regulate the filtration rate and the blood pressure. And so the first set of cells that we're going to talk about are granular cells. And these are located at the afferent arteriole. And again, the afferent arteriole is bringing blood into the glomerulus. So this is the afferent arteriole. And it's detecting blood pressure. So what kind of receptor does it act as if it's detecting stretch caused by blood pressure? Mechanoreceptors. So even though they're detecting blood pressure, they're not detecting that pressure specifically. What they're really detecting is the amount of stretch that's caused by that blood pressure. So if blood pressure is too low in that afferent arteriole, these granular cells are the ones that are releasing that renin. If you remember from this renin, angiotensin, aldosterone pathway we talked about with blood vessels and blood pressure, we said that when blood pressure drops, renin is released from the kidneys. Well, now you have to know that it's released from granular cells specifically from the kidneys. And those granular cells are right here surrounding that afferent arteriole so that they can detect that stretch. And when there's not enough stretch because blood pressure is too low, they release renin to trigger all of these pathways to try to increase blood pressure back to normal. So let's go through that whole pathway again. So this is your second time seeing this, You're going to have more questions on it. We actually will talk about it again in the very last chapter of this semester. So you should know that this is going to be probably seen at least a few times on your final exam. Okay. So when arterial pressure drops, it inhibits baroreceptors or mechanoreceptors, which is going to increase sympathetic nervous system activity, which is going to cause our granular cells to release renin from the kidneys. When renin is released from the kidneys, that renin is going to convert angiotensinogen, which is an inactive form of this enzyme, into angiotensin 1. That angiotensin 1 is going to be converted into another form called angiotensin 2, and that is done by this angiotensin converting enzyme. And which organ does that come from? The lungs. And then it is angiotensin 2 that's going to target all of these different pathways. So angiotensin is going to cause the adrenal cortex to release more aldosterone so that we're increasing sodium reabsorption, which is going to cause water to follow. So we have more water reabsorption, which increases blood volume and increases blood pressure. Angiotensin 2 is also going to target the posterior pituitary to release more ADH, which is going to target the kidneys to reabsorb more water, which also will increase blood volume and blood pressure. And remember, well, before we get there, also angiotensin 2 is going to cause an increase in thirst. So it's going to trigger your thirst mechanism in the hypothalamus, which will make your salivary glands work less so that your mouth gets dry. And then hopefully you listen to that cue and you drink more water. So it's going to cause you to intake more water, which is going to increase blood volume and then increase blood pressure. Remember that anything that changes blood volume is a long-term regulation. But what angiotensin 2 is also going to target is going to be your vessels and cause vasoconstriction in your arteries, which will also cause an increase in blood pressure. But it's not changing blood volume. All it's doing is causing vasoconstriction to increase blood pressure. So if we're just changing blood pressure without changing blood volume, this is a short-term regulation of blood pressure. Because we can cause vasoconstriction really fast, and as soon as they constrict, it's going to increase blood pressure. Versus the other pathways where we have to wait for a hormone to be released and then wait for that hormone to target the kidneys, allow the kidney cells to move that water and sodium, and then blood volume will increase and then blood pressure. That's going to take a lot longer than just vasoconstricting our vessels.
Now, all these happen at the same time, triggering the...
Yeah.
At the same time.
All at the same time, because that angiotensin 2 is going to be circulating throughout the body looking for all four of these different targets. Any questions? Okay. All right, so we just talked about granular cells, which are at the afferent arteriole over here. And then we have these macula densa cells. Macula densa cells are a group of cells at the ascending limb of the nephron loop. So these guys over here. And what they're going to do is they're going to be detecting levels of sodium. So if they're detecting levels of sodium, what kind of receptor are they going to be? Chemical or chemoreceptor. So they're going to act as chemoreceptors that detect sodium in the filtrate. And then we'll talk about how that's regulated in a second. So granular cells detect stretch and are going to help with regulating blood pressure. Macula densa cells are also going to help with blood pressure, but by detecting levels of sodium, and we'll talk about how they work in a few slides. And then granular cells are at the afferent arteriole, while macula densa cells are here at the ascending limb of the nephron loop. Your textbook will also talk about extraglomerular mesangial cells, but you can ignore those. We're not going to talk about those. All right, so you should know the three major processes of the urinary system. First, we're going to filtrate or do filtration where we filter things out of the blood. So filtration is going to produce a plasma-like filtrate because it ultimately is coming from the blood. We're filtering out the blood. Produces a plasma-like filtrate. Now, do you guys remember what kind of capillaries that we have at the kidneys? Fenestrated. Are those small holes or large holes? They're small holes because the large holes are the sinusoid capillaries. So fenestrated is going to be lots of small-ish pores compared to our sinusoid capillaries. So when we filter out the blood, the plasma-like filtrate that we end up with will have no red blood cells because those will not be able to fit through the fenestrations. Those holes are too small. Also, no protein. Because again, those fenestrations are too small to allow a whole protein to get through. Yeah.
And how is it that, like with certain medical conditions, you can, like urinate blood?
Oh, because so we're going to talk about later about like high blood pressure. So if you have like chronic hypertension and that blood is moving through the kidneys at a very high speed and high force, it can force red blood cells out and it can force protein out. But that's something that's wrong, essentially, because of the hypertension or because of damage to your kidneys or because of a massive infection in your kidneys. So you can end up with all those things in your blood, but that would be an abnormality or a pathophysiology of something going wrong. But somebody who's a healthy individual, they should have no red blood cells in their urine, no protein in their urine. If they do, it tells you that there's something wrong. Cirrhosis of the liver as well. Okay. All right. Reabsorption, again, this is where 99% of the filtrate is reabsorbed. And then secretion is about removing things from the blood that we don't want. So maybe there were things that were too big to fit through the fenestrations, but we don't want them in the blood. We want to get rid of them. So we'll secrete them out using the cells of the nephron to do it. So anything that didn't fit out through filtration that we do not want to keep will be secreted out at the last stage of urine formation. So secretion is getting rid of what we don't want. All right, so any questions about those three steps? Okay. So hydrostatic pressure forces water and small solutes from the glomerulus into the lumen or the capsular space. And so if you guys remember from like blood vessels and capillary dynamics, so hydrostatic pressure is essentially the pressure against the capillary walls that's caused ultimately by blood pressure. How fast is the blood moving through is going to cause that hydrostatic pressure. So hydrostatic pressure is going to force fluid out into this capsular space. So again, hydrostatic pressure, blood pressure ultimately is going to force water and small solutes from the glomerulus into the capsular space. Again, what kind of capillaries do we have at the kidneys? So the fenestrated capillaries, plus we have this extra filter on top. We have these cells called podocytes, which are like finger or foot-like cells that act as a second layer for filtration. I'll show you that in a second. These fenestrated capillaries plus the podocytes on top, they are permeable to water and small solutes, but not to blood cells or large molecules like proteins, like we said. So the holes are too small to let out red blood cells or proteins in healthy kidneys. And so this filtration part is non-selective and passive, which means that there's no proteins or anything like that that are going to be controlling the movement of solutes in and out. Okay, filtration is just whatever fits out is going to get out. Whatever fits through those holes will be filtered out. There's no selection during that. There'll be selection during the reabsorption and secretion portion, but this filtration portion is just whatever fits out of those holes is going to make it in the filtrate. And then we'll adjust after that. So this is the glomerulus with the fenestrated capillaries and the podocytes. So these cells, you see the nucleus here, these cells are the podocytes, where they have these little fingers that come out and they create a filter on top of the fenestrated capillaries. Little feet, yeah, like podiatrist feet, like that's what they're meant to be. So we have the small holes, which are the fenestrated capillaries, plus these slit-like filters on top. So anything that we filter out has to fit through the holes and through the slits that are created by the podocytes.
I'm sorry.
Say it again. So these are the feet of the podocytes, these things right here. So on here, you can see here's the fenestrated capillary underneath. So with all the little pores and fenestrations, and then these are the podocytes on top, creating an extra layer for filtration. So again, the solutes have to fit through the holes as well as fit through these slits to be filtered out.
Those are slits too, there,
Where?
that you just circled there.
Yeah, that's what I mean. So it had to fit through the holes and through the slits. All right, any questions about that? Okay. And so as you can see here, here's the blood plasma over here. Here are the fenestrations. These are the pores within the capillaries. And then here are the spaces created by these slits, are the podocytes. And so any solutes or fluid that gets filtered out has to fit through both of those pathways, through the pores and through the filtration slits. So plasma fluid moves through the filtration slits. Cells and proteins are too large to fit through. And so the filtrate resembles plasma without proteins. Any questions on that? Okay. Do you guys remember capillary dynamics and talking about hydrostatic pressure and osmotic pressure? Sure you do. With blood vessels, if you don't remember, go back, review. Your final is cumulative. So all this stuff is going to come back to haunt you. So we're going to talk about outward pressures versus inward pressures. So we have a hydrostatic pressure of the glomerulus, so HPGC is hydrostatic pressure of the glomerulus. And it's really the GC stands for glomerular capillaries, but it's hydrostatic pressure of the glomerulus, essentially glomerular blood pressure. How much force is pushing the blood through these capillaries? Now, all of the proteins and things should be held within the blood because we just said we cannot filter out proteins. So in a healthy individual, all that protein is still within the blood. So we have this osmotic pressure towards the capillaries. So OPGC is osmotic pressure of the glomerulus. So you'll notice that we only have osmotic pressure going towards the blood and not outward, because that pressure is going to be mostly towards the glomerulus, not out towards the filtrate, because most of the solutes are still in the blood. The proteins, all of that is still in the blood. Yeah. And then this hydrostatic pressure down here is hydrostatic pressure of the capsular space, because we'll have fluid in here from making that filtrate, from filtering out from the blood. And so there is going to be some outward pressure on the outside, from the fluid on the outside. So this is hydrostatic pressure of the capsular space. And so we're going to figure out what net filtration pressure is here. And it's just like what we did with the capillary dynamics. Outward pressures minus inward pressures. Our outward pressures here. Out pressures is 55 millimeters of mercury from this guy here. Our inward pressures are going to be 30 plus 15, which is 45. So 55 millimeters of mercury going out, 45 millimeters of mercury coming back in. So the net is 10 millimeters of mercury outward, which is going to allow us to filter out and make that fluid to start the diuresis or urine formation process. And this net filtration pressure is what is going to determine our GFR, our glomerular filtration rate, which is what we're going to talk about next. Now, am I going to make you memorize these numbers and ask you to do the math for me? No, but you should understand the outward pressures versus inward pressures and what allows us to make that urine or filtrate. It is these pressures that are working against each other. Any questions about this? So the bulk of the movement is going to be outward, which allows us to make the filtrate. Anybody still need these notes up? Okay. All right, so glomerular filtration rate, which is our GFR, it's the rate at which we make filtrate. So the kidneys make about 50 gallons of filtrate in 24 hours, but you're reabsorbing 99% of it, so you're not urinating 50 gallons of filtrate. That would be ridiculous. We reabsorb 99% of that, which are going to be the blue boxes on this pathway over here. That's all reabsorption. The 1% that's left over is concentrated waste, which will become our urine once we get out the collecting duct. And so secretion is epithelial cells that are lining the kidney tubules are going to secrete substances into the filtrate for removal. So reabsorption is all about keeping things that we want to keep. We reabsorb it back into the blood so the body keeps it. Secretion is all about getting rid of things that we don't want to keep. So we put it into the filtrate to be excreted out of the body. To remove the appropriate amount of waste, GFR must be about 125 milliliters per minute. If it's below that, waste can build up, which can eventually make us sick. So we need to stay at that 125 milliliters per minute. If you're lower than that 125 milliliters per minute, which can cause other symptoms. You have to get pretty low in order to need, we'll talk about dialysis later. Your GFR has to be all the way down to like 15. Yeah. All right, any questions about GFR so far? Okay. Review question. What specific tissue type is found in our kidney tubules? Simple cuboidal epithelium. Okay, so that's the kind of question that could end up coming back to you again. So just make sure you remember simple cuboidal epithelium makes up our kidney tubules. All right, so your kidneys have the ability to do intrinsic control. So it can maintain or regulate its own blood pressure from within without any communication with the brain or outside forces. Okay, it can control its own mechanisms. So one mechanism that it uses is called the myogenic mechanism. What kind of tissue do you think uses a myogenic mechanism? What is it? Muscle. Okay, myo for muscle. Now, it's going to be vascular muscle in our vessels. What specific type of muscle do we find in the vessels? Smooth muscle. So we'll use vascular smooth muscle. And what it's going to do, it's going to contract when those vessels are stretched and relax or dilate when it's not. And I'll explain to you why that is. Here we go. So if blood pressure is too high, we don't want blood pressure to be too high because what's going to end up happening is that filtration is going to happen too quickly. The filtrate is going to move through the nephron too quickly, and it's not going to give the cells enough time to reabsorb what it needs to reabsorb and secrete what it needs to secrete. So we don't want blood pressure too high because, again, we don't want the filtrate moving through the nephron too fast. So if blood pressure is too high, let me see if I have a picture of the... You can go back to here. Okay. So if blood pressure is too high, these vessels here, the afferent arterioles, are going to constrict so that it puts less blood through that glomerulus, less flow, so everything slows down and gives the nephron more time to do its job. Okay, so if blood pressure is too high, we don't want the filtrate to move too fast, so we just constrict that vessel so less blood flow goes through. It slows everything down, and then we will have more time to do that reabsorption and secretion. Okay, does that make sense? Okay. And then on the opposite side, if blood pressure is too low, then those afferent arterioles will dilate to increase blood flow and speed things up. Because again, we don't want to give the tubules too much time because they're going to reabsorb too much or secrete too much if it moves too slowly. And again, we have to keep our GFR at 125 milliliters per minute anyway. So if things slow down because blood pressure slows down, then we're going to need to dilate those vessels, bring in more blood, and try to speed up that process.
Professor, I'm so sorry. What did you say about the GFR at 135 what?
The GFR needs to stay at 125 milliliters per minute. So if blood pressure is too low and we slow that down, then we have to dilate the afferent arterioles that are coming to the glomerulus to speed up blood flow and to get things to move faster again. All right. So these intrinsic controls here, again, happen within the kidneys. Intrinsic controls are meant to maintain our net filtration pressure and our glomerular filtration rate so that the kidneys can do their job despite systemic blood pressure changes. So this myogenic mechanism lets the kidneys keep their GFR at 125, even if blood pressure is changing throughout the body. All right, questions about the myogenic mechanism? Okay, the next mechanism is macula densa cell feedback. So we said before that the macula densa cells detect levels of sodium. And so if sodium, and it's detecting levels of sodium in the filtrate. This is not in the blood, but in the filtrate. So if there is too much sodium in the filtrate, that means that there's not enough filtrate or not enough sodium in the blood. Okay? So again, if sodium is high in the filtrate, that means sodium is low in the blood. And so in order to combat that or regulate that, the arterioles are going to constrict, which means less blood into the glomerulus, which is going to slow down flow. And if we slow down flow, that gives the cells more time to reabsorb more sodium and put more sodium into the blood. We need sodium. Like, it's super important for neuron communication and all of that, maintaining membrane potentials, all of that. So in order to keep our nervous system working, we need an appropriate amount of sodium. And so if there's too much sodium in the filtrate, that means we're getting rid of too much. We want to be able to keep as much of that as we can. And so we'll constrict the arterioles to slow down flow, less blood flow into the glomerulus, which again gives the cells more time to reabsorb more of that sodium. Questions about that? Okay, on the opposite end, if we have not enough sodium in the filtrate, that means we have too much sodium in the blood. And so what we do is we dilate those afferent arterioles to speed up flow, to not give those cells as much time to reabsorb even more sodium. We want to push it through so that everything gets filtered out and a lot of it stays in the filtrate. Something should go through. All right, so that is macula densa cell feedback, also sometimes called tubuloglomerular feedback. So if you hear it, see that in your textbook, that's what it's referring to. But extrinsic controls. Extrinsic controls, which are happening outside the kidneys. Yeah. Yeah, go ahead. Okay. So extrinsic controls, again, everything happening outside the kidneys. This is to maintain systemic blood pressure. And you should already be familiar with all of these steps. So when blood pressure is too high, you have baroreceptors in your carotids and your aorta to detect that, which is going to send messages to the brain, which is going to cause the heart to slow down and arterioles to dilate, or arteries to dilate, so that we bring our blood pressure back down. Again, if our blood pressure is too low, those same baroreceptors will detect it, send messages to our brain to then constrict our blood vessels and increase our cardiac output in order to bring that blood pressure back up. So that's review. Make sure that you go over that again to remind yourself of the steps, because it pertains to here as well. All right. So our sympathetic nervous system, as you know, when your sympathetic nervous system is turned on, you have an increase in blood pressure body-wide. So what's going to happen is you're going to constrict blood flow to pretty much all of the organs of the body except for the areas that need it, right? You're going to be dilating the vessels that are leading to the brain, to the heart, to the lungs, and your skeletal muscle. Everywhere else is going to be constricted and receiving less blood, okay? So if a non-athlete does a marathon, somebody just gets up one morning and says, hey, I'm going to run the Bakersfield Marathon today. Don't do it. Don't recommend it. What's going to end up happening is that your afferent arterioles that are going towards your glomeruli at the kidneys are going to be constricted for more than 6 hours as you're running that marathon, which your body did not adjust to if you decide to just get up one morning and decide to run a marathon. This is why you should be training to run a marathon, increasing your length of time or length of your run a little by little over the course of weeks or months. Okay. So if your body is not used to restricted blood flow to your kidneys for that long, and you suddenly decide, I'm going to run a marathon, I'm going to have my sympathetic nervous system turned on for over 6 hours, what can end up happening is that you're getting less oxygen and nutrients to your kidneys that your kidneys are not used to already because you didn't train for it, and it can actually lead to renal failure. So you can actually die and experience renal failure because you decided to get up one day and decide to run a marathon and your body wasn't used to it.
You can also get hypoglycemia,
Mhm.
blood sugar
Yeah.
drops, and you black out.
Yep. So don't ever try to take on a really big activity like this, a strenuous workout or activity. Don't do a marathon or triathlon or anything like that without training for it first. Because your kidneys have to get used to having large amounts of time without adequate amount of oxygen and nutrients. Your body has to adapt to those changes. All right. There's an episode of How I Met Your Mother where Barney runs a marathon just one day, just says, hey, I could do a marathon. And then he does it, and then he goes on the subway, and then his legs don't work and he can't get up. And so he ends up taking the whole subway around, and he can't go anywhere. It's funny. All right, so remember that it's going to be the granular cells in the afferent arterioles that are going to be regulating blood flow here. So if blood pressure is too high, they constrict to allow for less blood flow. If blood pressure is too low, they will dilate to allow for more blood flow. And that's filtration. Okay? So step 2 of diuresis or urine formation is reabsorption. So the first step, again, is filtration that we just talked about, filtering things out, which is non-selective. Anything that fits out is going to be shipped out into the capsular space. And then the next step is all about reabsorbing the things we want to recapture. Things that got filtered out that we don't want to get rid of, we don't want to excrete, and we want to keep, will be reabsorbed back into the blood. So, which again is going to happen, if we're talking about solutes, it's going to mostly happen at those peritubular capillaries, at the cortical nephrons. If it's water, it's going to be mostly at the vasa recta with the juxtamedullary nephrons. But this is why we need those capillaries around the tubules so that we can put things back into the blood or take things out of the blood when we need it. So there's 2 ways that we reabsorb things. We either use the transcellular route, where we transport things across the cell, either completely across the cell from the apical side to the basilar side, or it'll go through the apical surface and then to the intercellular space. But again, transcellular route, it goes across the cell, okay? Or we can use the paracellular route, which goes in between cells. That was not a good color to use. So that's all you have to know about that. Okay, so how we reabsorb things either across the cell, which is called transcellular, or in between cells, which is called paracellular. All right, questions about that? All right. Now this is a little bit more complicated here. So we talked about how we reabsorb sodium is through a sodium-potassium pump. Okay? When sodium is moving in one direction, potassium is moving in the opposite direction. Okay? We're going to be using ATP-ACE pumps to do this, so it requires energy to do this. It requires energy to use a sodium-potassium pump, and it's going to cause energy to use a co-transporter that we'll talk about over here. So that's one mechanism that we use. Remember that the more concentrated that we make the blood, water is going to want to follow in that direction. Water always wants to follow the more concentrated side of any membrane. So as we're moving sodium into the blood or other solutes into the blood, water is going to follow. Anybody remember the channels that water uses for transport? If it's not moving by simple diffusion, it uses very specific protein channels. Starts with an A. They're called aquaporins. So if you didn't learn that in Bio 32, make sure you know it now. They're called aquaporins. Those are the water channels that we use. So water can move by simple diffusion, but if we need to move bigger amounts of water, we use these water channels called aquaporins. People that are pregnant make more aquaporins, and so they end up retaining more water. That's why you get the swollen hands and feet by the time you get to the end of it. Lipids are going to move by simple diffusion. You should know lipids can cross the membrane on their own. So anytime we have to absorb lipids, it's going to go through that mechanism, simple diffusion. And then we have these co-transporters. What a co-transporter is that in order to reabsorb solutes like glucose, amino acids, ions, vitamins, we're going to use a co-transporter, which means that every time we move one of those items, we move sodium with it. It's called a sodium co-transporter. So we're moving sodium and one of these different solutes, either glucose or amino acids or vitamins or ions. It's a co-transporter. So it has to have sodium every time we reabsorb one of those solutes. Questions about that? Okay.
In the notes, it said the passive diffusion, but over here it's saying passive diffusion.
Let me see. Oh, passive is the same thing. Passive diffusion, simple, it's the same thing. Simple diffusion is a mode of passive transport. Okay. Now, we do have what are called transport maximums, okay, where we can only reabsorb a certain amount of each solute. And then once we reach that transport maximum, those proteins will no longer transport that solute until we drop below that transport maximum and can do it again. Okay, so there's a maximum to how much we can reabsorb. So the apical surface of epithelial cells lining the nephron tubes have a maximum rate of transport, or with what we call Tmax. So this is what happens with people who are hyperglycemic or have diabetes. So what ends up happening is that they have so much glucose in their blood. If we go back to that picture of the nephron, we can do it here. So they have so much glucose in their blood, right? A lot of it gets filtered out, so much glucose, okay, if they're diabetic and not being treated for it. So then, we need glucose to make our brain run and all of that. And so what ends up happening is that we reabsorb all this glucose into the blood, but then we reach a T maximum where we cannot reabsorb any more. And what you end up with is lots of glucose in your urine because you've reached that T max. And that's one of the markers that we can use to diagnose diabetes is glucose in the urine. Because you shouldn't, as a healthy individual, if you have the right amount of glucose in your blood, you should be able to reabsorb all of that glucose and not have any left over in your urine. If you have leftover in your urine, that's because you have too much glucose in your blood, you reached a T maximum and you couldn't reabsorb any more. And so you end up having leftover in the urine. Right? Does that make sense? We're going to have a lab activity on that, and we're going to talk about that a little bit more in lab today. All right. So there is a newer diabetic control mechanism called a sodium glucose co-transporter inhibitor. And so what it does, it decreases the Tmax for glucose so that we can't reabsorb so much of that glucose into the blood. And so what would be the results in the urine? Well, we'd have more or less glucose in the urine if we lowered that Tmax. More, right? Because we're going to reach that Tmax sooner. And what about in the blood? Will we have less or more? Less. If we reach that Tmax sooner, we're reabsorbing less, right? So we're going to have more glucose in the urine, less in the blood. Why are patients taking this drug at an increased risk of death from ketoacidosis? Okay, the reason for that is because there's less glucose available in the blood. And if you remember, if we don't have enough glucose available for the brain, that acetyl-CoA can't move on to the next step of cellular respiration, and it builds up, and then we convert those to ketones or ketone bodies, which are acids. So now we have acids building up in our blood, which can end up leading to metabolic or ketoacidosis, which can depress your nervous system and eventually lead to a coma or death. Questions about that? Okay. I think this is like pretty much like the end, I think. So we're just a really quick review, and we're going to come back to this when we come back. But reabsorption is going to happen at the proximal convoluted tubule. There, at the proximal convoluted tubule, we have something called obligatory reabsorption. So 65% of our filtrate has to be reabsorbed at the proximal convoluted tubule. So we said that we reabsorb 99% of what gets filtered out. 65% of that 99% has to happen in the very first part. All glucose and all amino acids are reabsorbed in that very first part. We should not be reabsorbing any of those things later on in the nephron. It has to all happen at the proximal. And then at the nephron loop, that's what's going to be responsible for making dilute or concentrated urine by countercurrent multiplication, which we're not going to talk about. You don't have to worry about that. But just know that at the nephron loop is where we're making adjustments to water. And the remaining parts of the reabsorption are going to happen under hormonal control at the DCT and the collecting duct. So for ADH, know that water is being absorbed through aquaporins, those water channels we just talked about. Aldosterone is sodium reabsorption, which is going to happen through sodium-potassium pumps. AMP, if you remember, is an antagonist to aldosterone, so it inhibits sodium reabsorption, so it's just going to slow down those sodium-potassium pumps. And then PTH is for calcium, and then we just reabsorb that calcium through cells. We won't talk about the exact mechanism. All right. And so when we come back, we'll review more of the renin-angiotensin aldosterone pathway and talk about secretion.